This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 31 for May 8th, 2009. From New York City, I'm Vincent Racaniello.
I'm Dick Despommier.
And from Western Massachusetts, I'm Alan Dove.
And joining us today is Marc Pelletier from Futures in Biotech. Welcome to TWiV, Marc.
Thank you very much for the invitation. I really appreciate it.
Where are you actually physically located, Marc? I'm
in Cleveland right now, just outside of Case Western Reserve University, right outside the med school. Yeah.
We know that you're the head honcho at Futures in Biotech, which is a TWiT network podcast about science. I was lucky enough to join you a few times on that podcast. And so you are the science podcast maven, I guess.
Well, you know, the point of doing the podcast was we started early in 2006, and I was doing postdoc at Yale, and all these great scientists were stopping by and giving talks. And at the end of every talk, I noticed, you know, I'd raise my hand to ask my question, and then there'd always be this line of questioning that would follow, and you know, everybody in the audience would just sit and pay attention. And you know, we're talking some fantastic scientists, you know, world class scientists, and we're having these conversations in front of the group, but nobody else would interject or stop or raise their hand, and they were a lot of fun. So at one point, when Leo said he'd like to do a biotech podcast, I gave him an email, and of course, coming from Yale, where you know that's his alma mater, so he holds it dear to his heart. He responded right away, and I got a phone call in the lab from, you know, this is the voice in the box, right? So anybody who listens to a number of podcasts, there's bound to be a Leo Laporte podcast in there.
Sure,
right.
Oh, that's a great story. I didn't realize that. Well, I did a Futures in Biotech with you last month with Peter Pelletier on flu, and it was just perfect timing because a few weeks later, all hell breaks loose with
that
influenza.
Was scary,
Wasn't
right?
That amazing? Yeah.
The time. Are we the cause of the media hype?
I don't know. You asked me, who should we get? Let's get a viral. So you and I had corresponded because we picked Futures in Biotech very early on as a pick of the week. So that hooked us together, and that's what I always hope would happen when I make picks, is that we hook up. But you're about the only one that's done it, Marc. And then you said, let's do a virology futures, and I said, Peter Pelletier, flu is the hot thing. So we did, and now, well, there you go.
Yeah, that was episode 40, by the way. And I'd also like to mention that you were a guest on episode 42, and you helped really clarify what was going on with swine flu. So I really appreciate you coming on to do that one.
That's twit.tv/FIB, is that right? That's
right, twit.tv/FIB, and episode 40 and 42. So those are the two ones.
So 42 was interesting because we did that live. That went through the TWiT network. And that was a lot of fun, you and I talking live. And then it was, of course, recorded, so that's now episode 42. I think, Marc, that is the future of science teaching, that sort of webcast.
Well, this is really long tail, right, in terms of media production. And I think, you know, because of the fact that it's very much talk radio that people go get on their own with their own interest, that long tail becomes extremely large. And I think for example, doing some crossover like today, you can really expand that audience, right. Your audience, and also introduce your audience to some great shows.
Sure.
And where, you know, how long is it? You know, there was never these great science radio shows, right?
No, never. I mean, there is always some kind of science coverage, which is not very satisfying, but something dedicated to science, no. So you're probably among the first. And now there are some, but, you know, there's not a lot of hard science, I would call it. There's a lot of medicine, there's a lot of nursing, EMT, which is great, but hard science like biology, virology, there's not a lot. There are courses, there are podcasts for courses, but in terms of this sort of thing, interviews and conversations, there's not much. So
we actually threatened to insert a basic principle of virology into each one of our podcasts, and we have yet to start to do that, but I would encourage us to think along those lines.
No, I agree. I think we should do that.
Like cover the attachment phase of viruses entering cells.
There's so many things we can do.
Yep.
In fact, today we're going to make a change. And that is, gentlemen, I'm going to ask you for science picks, and they can be anything having to do with science. It can be a book, a podcast, a website,
Television
a museum, television show, anything. But today we have usual virology stories, and we're going to start with influenza because the news is dominated still by influenza. People are fascinated by this virus. But let's go over the numbers before we just talk about a few things. U.S., as of this morning on the CDC site, 896 laboratory-confirmed cases, 2 deaths, and this is 41 states. So almost every state now has a virus, this virus of some kind. Of course, this is the new H1N1. Morbidity and mortality weekly report just came out with an update on May 8th. And it looks like they have a graph of the cases in Mexico. And it looks like even in middle March, they had some suspected cases. There were low levels of suspected cases with this virus, and then it took off at the end of April. So this virus may have been around for a while. So in Mexico, 822 confirmed cases, 42 deaths. The interesting thing there is that there are almost 12,000 suspected cases. Now, there are only 822 lab-confirmed cases. Suspected is a very broad definition. It's basically fever, sore throat, and coughing, which could be flu. It could be...
All my students have that.
Yeah, it could be the common cold.
That's right.
Especially during finals, yeah.
Exactly.
In fact, I was talking to...
And I'm their headache.
I was talking to someone yesterday who's been involved in sample collection here in New York, and he said that, in fact, some of them are rhinoviruses that they're isolating because they have people with cough, sore throat, and fever. It's a very broad definition.
But no headache. I think they threw headache in there as part of the
Well,
syndrome.
here on the CDC site, it says the case definition, fever, cough, sore throat. That's it.
Really?
No headache.
Okay.
So how do they confirm the case? Maybe you could tell us how they do that.
So according to the CDC site, they do real-time reverse transcription PCR or viral culture, either one. So they can take a nasal swab or a throat wash, do RT-PCR. And actually, you can go to the WHO website and find the primers that you need to do this. So we could do it ourselves. You have a PCR machine, Mark?
Absolutely. Every bench should have one. Every bench.
Every home should have one.
Right.
I think Alan has a PCR machine.
Well, I think you can buy the outdated ones on eBay now for a couple $100. So.
Right.
Yeah. I when I started in this one lab at Yale, I spent 3 or 4 weeks just trying to amplify these 12 genes, and it was like 1 a week or 2 a week would pop up. And I realized that I was using a crappy thermal cycler, right? So beware of the eBay ones. Get them verified. Do the controls.
Right. No, I'm saying for the home user, it might be.
Right.
So
the garage lab, right?
Yes.
The PCR, and then viral culture is harder because it takes longer, and you need to know what you're doing. But
Do not try this at home.
You put this nasal swab in a cell culture, a susceptible cell, and you wait for the virus to grow, and it grows. It's growing because it kills the cells. Cells round up and detach, and then you take that virus and you can do you could do RT-PCR on that directly. You could do serology and confirm it.
You could have a susceptible lab tech snort it.
You could do that too. In the case of this H1N1, it seems to be in general pretty mild. That's most of the cases, except for Mexico, are really mild.
Right.
Now, when you do some number crunching here, if you take 42 patients and you divide it by 800 and some confirmed cases, you get a fatality rate of 4%, which is pretty high for flu. Like 1918 was 2.5%.
But the age group was specific.
Yeah, but Peter Pelletier said it's just too early to make any conclusions. The numbers aren't there yet. Mm-hmm. But if you put the 42 deaths over 12,000 suspected cases, then you get 0.4%, and that's more in line with seasonal mortality. So, the sequences of these viruses have shown that they're no different from any of the other isolates anywhere in the world. So it's not clear what is going on yet in Mexico.
And clarifying something that we talked about before, the Mexican sequences have in fact been released. We're just not on the site.
So actually, they were released on the 24th, and they were released at a site called GISAID, the Global Initiative for Sharing Avian Influenza Data, and you have to register and log into that. But they were there, and I just didn't look there. I actually got a membership just last week, and they're all there. It's wonderful. There are some at NCBI also, and they're all quite similar, and there's nothing that sticks out as being odd amongst those. So we don't know why that we have this deaths in Mexico, and it'll it'll be a while. One of the interesting aspects of this, well, maybe we'll talk about it later. There's a protein called PB1F2, which is encoded in a second open reading frame. And we have an email about that later, so maybe I'll hold off on that. But according to Peter Pelletier, he doesn't see any reason to think why this is terribly virulent, and he says there's about a 50-50 chance that it will never go anywhere. So it will either burn out or it will continue. But he says it could continue, and that the evidence is if it starts to grow or cause outbreaks in the southern hemisphere in the next couple months, which is when their winter is. Then if that happens, he said it'll be back here in the fall, and then we should have a vaccine ready. But if there's nothing in the southern hemisphere, then that's probably the end of it. And we don't know which way it's going to go, but in a few weeks we should know.
How do these animal ones form? This is a quick question. Is it a person who has a co-infection of two similar viruses and there's a recombination that happens?
Well, Peter thinks it could be. I mean, everyone thinks this happened in a pig, right? So two different viruses co-infect a pig, and then a pig worker gets it and spreads it. But this virus has never been found in a pig, and what makes this virus unusual is that it has a combination of genes from pig influenza viruses from North America and from Eurasia. And that particular combination hasn't been seen in pigs. So that could have happened in a pig, or it could happen in a person who's working with a pig and happens to be infected with two viruses. And the problem is, even if you do find this or a similar virus in pigs, it doesn't mean it originated there. It's very difficult.
There's another thing to keep in mind here, gentlemen, and that is that there are about, I wouldn't say how many varieties of pigs, but there are tremendous numbers of inbred strains and outbred strains of pigs. And pigs are being shipped all over the world from place to place. So to know how this actually started or where it actually started would require you keeping track of pigs. For the most part, United States pigs are not earmarked. They're not tattooed. And for that reason alone, you can't find out where they came from. You don't even know whose farms they came from in many cases. I used to work on a disease called trichinosis, so I know.
You know pigs, huh?
Pigs, I know pigs from start to finish, and it's a very, very difficult subject area.
I like the finish part.
Although I should point out at this point, one of my predictions from the last show was quite true. I went to the supermarket right after we recorded that show, and in fact, all of the pork products were deeply discounted.
Yep.
Oh.
Yep.
And you can't catch it like that anyway.
I know, but hey, I'll exploit the situation. I totally stocked up on pork chops and been eating barbecue. It's great.
I hope you thoroughly froze it. That's all I can say.
Yeah, I froze it and then cooked it.
All right.
So, but we should just give the international numbers as of this morning on WHO's site. 24 countries, 2,384 cases so far. And over almost half of those are in Mexico. So that's a big outbreak. Now, they have a very nice map on the WHO site, which shows you where all the cases are. And they're conspicuously absent from the southern hemisphere.
Yep.
There are just a few in New Zealand, 5 cases, Colombia. Dick, where's the equator here, roughly?
It's where Belém is. It's way down.
Oh, it's way down there. Colombia is in the northern hemisphere.
Absolutely.
El Salvador, Costa Rica, Guatemala, Mexico, of course.
Yep.
So only Israel's in the northern hemisphere.
Oh, of course.
Korea, China.
Yep.
So it's just New Zealand confirmed cases, so not much in the southern hemisphere. Right. But I think it, as Peter said, 50-50 chance it's going to take off. We don't know. And as Peter said, you can't predict the future, especially with viruses.
It's even hard to predict the past in some cases.
As you get older, you can't remember it either.
That's right.
I do have a question.
Of course.
How come they couldn't contain it?
Mexico,
to Mexico.
You mean?
Yeah, Mexico. Well, I know it's a populous city of 10 or 15 million or more.
But it didn't just arise there. It arose in several regions of Mexico. And I think by the time they realized what they had on their hands, people had already gone out of the country.
Yeah, I think that's, if you look at the graph on MMWR, there are suspected cases in the beginning, almost halfway through March,
right?
Which is way before we had any idea that it was a different virus. So it could have started this. That's, I think, what happened. It was spreading long before we knew about it.
My guess is that they wouldn't have even known that if those children hadn't died.
Yeah,
that did raise a red flag at that point.
Well, what about does this tell us something about viruses, our ability to contain viruses?
Yes, it does, actually.
Yes, quite.
That is, we can't.
All right.
But what we can do is go on red alert once we know one is loose. And that's what we do very, very well now. And that's why this show is so riveting, because The fact is that 20 years ago, you couldn't have done this. You couldn't have had a TWiV. You couldn't have had a worldwide network that WHO was in charge of that works as effective as this does. And I'm actually amazed at how widely read ProMed is and PubMed and all those other kinds of publications that have alerted virtually every region of the world as to what's going on. In a moment's notice, you can tell when there's an outbreak.
Yeah, and the technology on this has not only come a long way in the past 20 years, it's evolving very rapidly now.
That's right.
To
Cell
the point,
phones.
well, right, cell phones and also the diagnostics that we have.
Sure.
Yes, it was in this case. In this case, it was impossible for Mexico to have discovered this and acted on it within the time frame of a virus spreading. But one could easily imagine, 10, 15 years from now, if we get to a point where you've got some of these chip-based methods, if they actually live up to 20% of their promise, right. You could imagine a swab in a doctor's office confirming that this is not any known type of influenza, and that would raise an alert.
No question.
So the surveillance could get to the point where we might be able to contain these things even better.
That's right.
But in this case, it would have, by the time they found it, all the measures that people were proposing were just closing the barn door after the horses had left.
The thing is that there's an incubation period here, which a lot of these other more serious virus infections like Ebola, they have a more rapid fulmination. So there's a lag time between you catch it and the time you exhibit symptoms, and that's what gives flu its chance to get out of the zone.
Right.
Whereas Ebola will never get out of that zone, basically, as long as there's an outbreak that you can identify.
I think it probably made the measures that we took probably made some difference in containing it. But as you say, you can't completely contain it. Quarantina, right, Dick?
Exactly right.
One thing that Peter Pelletier did say to me was that we should be making a vaccine, and there are a number of companies who are doing this. And in fact, I think it was USA Today that had a really nice graphic on flu vaccine production, which we'll put a link to. Also has a nice photograph of this virus, an EM, which was taken at CDC. Unfortunately, they colored it blue.
Oh, well.
And as we know, viruses don't have color.
Rhymes with flu.
They don't have color.
You only get
green
blue
and slimy.
That's right, green and slimy.
Green and slimy. Remind me to mention that later. My pick has to do with green and slimy.
That's Pseudomonas aeruginosa. Come on, guys.
So flu vaccine production. It's a nice timeline. USA Today is not so bad. I spoke to a reporter last weekend, a science writer. He was pretty good. So this graphic shows that from January through May, what happens is the flu strains for the season are selected. The samples are distributed to manufacturers. It's grown in eggs, harvested, and all that takes until May. Then from June to July, it's tested by the FDA. The strains are blended, and then FDA licenses it. In August, the vaccine is put into vials, shipped in September, and then in October, you start immunization. So we are just starting here in May, so we are going to be a bit later than October, I think. But this timeline is very nice to show you what's involved here and why it might not be ready exactly in the fall.
Do you think some other approach could be used, like antisense RNA, or antisense little segments of antisense DNA, I suppose, that you could take as a nasal spray?
People are working on that, sure. They, in cell culture models, they use these siRNAs is what's being done now. They're effective at inhibiting viral replication. The key is how to deliver them, because they are, 21 nucleotide duplexes. It may be that the most effective way would be in a virus vector. So maybe an adenovirus vector delivered intranasally would be effective for that. But that certainly is being worked on. That's the next generation antiviral approaches for sure.
Because those would be faster to make, right? Vaccines take time to grow.
Oh, yeah. Yep. You have a sequence and that's it, right?
Yeah.
And we talked on TWiV a while ago, gentlemen, if you remember, a trial for an HIV. It was actually a ribozyme, I believe, but it was the same idea, put into stem cells. And that's very effective at preventing viral replication. So I'm sure we'll see more of these in the future as they come out. One last thing I want to touch on with respect to influenza. I talked with Peter about this. This is very interesting. So in the past, whenever we have a pandemic strain, the old strains go away. So in 1957, the H2N2 viruses replaced the H1N1s, and the H1N1 disappeared. In 1968, the H3N2 strains came on. It's the new pandemic strain, and replaced the H2N2s. And we don't really understand why that happens, but Peter thought that we might have an unprecedented event in the fall, 4 different strains circulating. H3N2.
H5N1.
No, it doesn't really circulate in people.
No.
2 different H1N1s,
all right.
The Russian H1N1 and this new one, and then influenza B virus.
Ah.
All these things we learn get thrown out the door.
Well, we
right?
relearn. You have to relearn.
When I was taught influenza, it was a new pandemic strain replaces the old one. And so here I am going into this situation with that idea, but he tells me it may not be the case. So it will be interesting to see what happens.
Also, the human dynamic is totally different today. I think we pointed that out last week also. The number of people that were actually alive in 1918 versus the number that are alive today, the modes of transportation, the availability of healthcare systems, all these things impact greatly on these issues. And it's impossible to predict based on the past, where these things didn't exist, as to what's going to happen now that they do exist.
Yeah, that's right. I mean, in 1968, the last pandemic, agriculture and growing animals for food was very different, right?
Still, you're absolutely right.
Yeah.
I mean, now they're growing pigs almost hermetically. If you go to Iowa, they have these piggeries that are absolutely round, contained pig farms that the corn goes in and the pig goes out and nothing else happens. All the excrement is taken care of, methane digestion, energy generation, all kinds of weird stuff.
Well, the excrement is accumulated in large lagoons, right?
No, that's in North Carolina. In Iowa,
right.
That's turned back into methane digesters, and the methane is then burned and used to generate electricity for the piggery.
Those clever Iowegians.
They got the idea from Denmark, actually, from Denmark. But you're right. I mean, they are clever people, if they have to be.
Can't you grow bacon in cell culture? I think you can.
Well, you can, but
You want your bacon, you're going to get flu. That's the bottom line, at least for now. I found out, actually, that they do immunize pigs on commercial farms against flu.
Yeah.
It turns out that when pigs get sick, it's for about 3 to 7 days, but they don't eat for 2 weeks. And that's not good for their weight. Right?
So, Vince, let me ask you a question for a change. What's the difference between raising cows for a meat source and raising pigs for a meat source? Why do you think so many people raise pigs and not a lot of people raise cows? That's what
pigs are more efficient at turning vegetables
to
food
Vince.
into
Alan, you stay out of this.
Alan knows.
You already know this answer. I'm trying to catch you on something. Come on.
Because pigs grow quicker to market size, right?
They do. It takes 6 months.
Yeah.
And you can grow a pig.
How long does a cow take?
18 months.
How many babies can a pig produce?
Oh, lots. Dozens.
Every 6 months, like 18 or something?
Something like that. Cattle, one at a time.
What's the difference between a pig and a swine and a hog, Dick? Nothing?
Nah. If you let the domestic pigs loose, they revert back to wild pigs.
So actually, this is an interesting question. Someone asked me yesterday if wild pigs get influenza. And I didn't know the answer. But there's not much. Do you think they do?
Of course.
Of course.
Absolutely. Why wouldn't they?
Where would they get it from?
Hunters? I don't know. Other pigs? Maybe pigs are constantly getting loose. If you travel throughout the world and you look at these isolated islands out in the Pacific, they're all overrun with pigs or goats or donkeys or some other released animals that these shippers put on the island so they'll have available meat sources when they return to those sites.
Okay. So they must. It'd be an interesting thing to look at, to go out and try and isolate influenza virus from wild pigs. Personally, I'm going to focus on mice. It's just easier to track them.
They get it from the birds, the migrating birds, the wild
migrating birds. Sure, absolutely. But before 1918, there was no flu in pigs, as far as we know.
Oh well, we didn't look.
But we didn't look, probably.
How would we know?
But until 1998, the same strain was in pigs, at least commercial pigs, for many, many years. So, but again, you're right. It's a matter of.
And I think
why do you question this?
Before 1916, we didn't do a lot of traveling out there. I mean, we didn't know about the rest of the world. We had a Monroe Doctrine of just closed doors, and that was it. The moment you have a war, of course, everything changes.
Why do I question there being viruses in wild pigs?
Why wouldn't there be?
I have no good reason for there not being, but it just hasn't been found. Or if it has been, I'm not aware of it, but I'll have to look.
No, the wildlife biologist would know the
Hey,
answer.
Alan, do a PubMed search as we give our next commercial advertisement.
I bet you.
All right.
I bet you it has been found already.
Yeah, okay, we'll let Alan do that. But now we have a word from Citrix, the makers of GoToMyPC.
Aha.
Now, I rely on my office computer here. Do you, Dick?
I do.
And Mark must also. And if I were using GoToMyPC, I could access it from anywhere in the world.
Really?
So I don't have to come here. If I travel, I can always access it. If I forget a file, I can always access it.
That's great.
And you can do that with GoToMyPC, which is basically like a remote access program. You install it very quickly and easily on your computer, and then you can reach it from anywhere.
Sounds great.
So you can reach your office PC from home. It's very easy. You log on to gotomypc.com, and your desktop appears there.
Is it very expensive?
You can use any program. No, it's not at all, Dick.
Really?
It's reasonable, highly reasonable.
Oh, but it's reasonable price. That's all we can
Reasonably
say.
priced. You can use any program, work on any file, check email, access your network. It can be a life-changing solution for you.
My goodness.
And right now, you can try GoToMyPC free for 30 days.
See?
But
That's
you must
very reasonable.
Visit gotomypc.com/podcast. Gotomypc.com/podcast. You get a free 30-day trial.
Great.
And tell them TWiV sent you. Devin, it's my son. Alan, sorry. Alan,
That wasn't even a Freudian slip.
Oh, God. Well, Alan was my student, so, there's sort of a...
That's true. That is true.
Right.
Did you find...
Yes, so I immediately found some hits, and from the Journal of Wildlife Disease, here's a fairly typical one, influenza exposure in United States feral swine populations.
Yep.
So these folks are at the USDA and APHIS Wildlife Services in Fort Collins. Mm-hmm. They found that they did a survey in U.S. feral pigs, and they found pretty widespread seropositivity to different serotypes. So let's see. All seropositive swine were exposed to the H3N2 subtype, which is the predominant subtype circulating in domestic swine. But in San Saba County, Texas, they had 15 seropositive samples. 4 were positive for H1N1, 7 for both H1N1 and H3N2.
Look at that.
And they go on to analyze the data some other ways, but it's the highest number they saw was 14.4% of the sampled feral pigs in Texas were seropositive. Mm-hmm. So it's not it doesn't necessarily always track what's circulating in domestic swine.
Right.
So they seem to have other sources.
Any Australian literature on this? There's a lot of feral pigs down in Australia.
Yeah, I'm not immediately seeing. Looks like a Chinese paper.
One of the estimates I've seen, by the way, for Australia is that they've got over 3 million
feral pigs.
Wow,
that's a lot of pigs.
How many are in New York City, Dick?
None. We don't have a single feral pig wandering the streets of New York. It's all domestic pig here.
There's a really nice zoo.
That's right.
Yes,
there is.
That's right.
There is a very nice zoo.
While life can be boring, we don't have a lot of feral pigs.
They have pigs in the zoo. Okay, well, I think that's enough pig discussion today. Actually, I had a student defend her PhD thesis yesterday, Jennifer.
Yes. Oh, good.
And her dad
yes.
used to work at a hog farm in Iowa.
Really?
Oh.
So that gave me a nice introduction for her yesterday.
Interesting. Can you take an aside right now?
I could, sure.
We had a Trichonella meeting once up in Canada, and there was a stuffed wild pig, and there was a contest to see who could name this pig with the funniest name. And there was a lot, and the reward was a very fine bottle of French wine, and the winner was Boar George. That was the name that they selected. It's kind of a funny name, but in the intervening years, I've thought, why didn't I submit a name for this? And you'll never guess which one I would have submitted.
Despommier.
No, but we can have a contest with TWiV to see who can come up with the most clever name for a stuffed wild boar, and I'll give you the name that I would have put in now.
Okay.
Pygmalion.
Excellent.
I think I would have won that wine. I didn't win the wine, but the other was Boris. Someone actually put that name in as well, but Boar George actually won the contest.
Boar George is great. That is a good.
It's a good.
That's really good. I like that.
Yeah.
Now, listen, I'm going to follow this rat hole a little further. Okay.
Go ahead.
When Mark and I did this live Futures in Biotech last week, Colleen, who was running the boards for us, the audio board, showed a video of a sneezing panda. So we made that the name of the episode, Sneezing Panda-emic.
Sneezing pandemic. Pandemic. Oh, yeah. You remember what I wrote you? I said we're making much too much out of this. In fact, we're making much a chew out of nothing.
Oh, God, all right. That's it for the rat hole.
Okay, I got one last scientific rat hole.
Sure.
So, first question is, what is the viral receptor for influenza?
Cyalic acid.
Right.
Cyalic acid. So this is it on the cell surface?
Oh, yeah, it's on just about every cell, yeah.
That's what the...
Cyalic acid. It's not a protein, eh?
Well, it's cyalic acid linked to a protein. Yeah, it's
which protein?
Can be any protein. Doesn't matter.
It's all over the place.
It's too bad. It'd be nice if there was where you could engineer a pig to not express the protein that's the viral receptor.
Of course.
Yes.
And then have a super pig.
Sure.
That's right.
Yeah, you make some kind of transgenic knockout pig, right? But unfortunately, it's ubiquitous, so that's out of the question.
It's sort of like making this is the rat hole aspect of it. If you could make a prion-free cow, you'd have a mad cow-free industry, which would be worth billions.
Absolutely.
All right.
That may be possible, but...
just call it angry cow disease from now on.
That could be possible. Okay, I found a couple of non-influenza virus stories, just to let our listeners know that there are other viruses out there.
It's not this week in influenza.
Not this week in influenza. It's the news, which I used to look for virus stories, had nothing on other than flu for the past 2 weeks. It's amazing.
Yep.
But I found two articles which I want to talk about. They both have to do with parvoviruses, which I don't believe we have spoken much about here. And the first one has to do with dogs. And if you have a dog, you know that one thing you have to immunize them against is canine parvovirus. You have a dog, Dick?
Nope.
What about you, Mark?
I used to.
Used to have a dog, but it died of canine parvovirus infection?
No, eating too much food.
Yeah. Parvovirus are rather small DNA viruses. They're non-enveloped icosahedral viruses, single-stranded DNA genomes. They're circular, and they're both human and animal pathogens. And the canine parvoviruses, well, I think there was an article on Oprah about Oprah's dogs dying a few weeks ago from canine parvovirus.
Really?
It can be a lethal infection.
Does it have a common name?
No, it's called canine parvovirus infection. Yeah. So, for example, I picked up a story here from, where is this? Pomona. Dozens of dogs had to be euthanized at an animal shelter after becoming infected with parvovirus. Yeah? So they actually call it canine parvovirus. It's highly contagious, attacks the lining of the digestive system, and prevents the animal from absorbing liquids and nutrients. They can die of dehydration, especially puppies. So that's why you get a vaccine for this virus. And every now and then you will see these stories about canine parvovirus infection. Now, there are a bunch of interesting viruses in this family, which is called Parvoviridi. There are human viruses, there are dog viruses, cat viruses, minks, pigs, rodents. And the interesting thing about the canine parvovirus is that it used to be a cat virus that underwent a host shift. And the story of this we will not talk about today because there is a scientist up at Cornell, Colin Parrish, who had worked on this story, this host switch between cats and dogs.
Wow.
And it involves basically receptor changing, residues on the capsid that recognize receptors. So now this virus jumped from cats into dogs, and it's rampant in the dog population.
Is he at the Baker Institute by any chance?
He is, yes.
Do you know
Are we going to get him on the show?
Do you know what that's famous for?
I would love to get him on the show. He's really got this whole story wrapped up. I hear him tell it. He tells it beautifully. And I will email him after this episode to get him on the show to tell us the story.
I think the Baker Institute's main fame is that they invented the distemper virus vaccine.
I think it's the Boyce Thompson Research Institute, not Baker, which is specifically for dog diseases, I think. But I may be getting this wrong. And maybe there are people who work there who are listening and cringing. I'm sorry. But we'll get Colin on to sort it out. I'm sure he'd love to do it.
I used to do some collaborative research with some people up at Baker.
Yeah. I don't think it's Baker. I think it's Boyce, because I did visit it once.
Okay.
It's very interesting. But these parvoviruses are quite interesting. The canine switch is very interesting, and I want Colin to tell you about it. The canine virus emerged in 1978 as a host range variant.
Wow.
And it can still go back into cats. But they infect by the oronasal route. They replicate in the pharynx, and then they spread to lymphoid tissues. That infects multiple organs, thymus, lymph nodes, spleen, intestine. And that's where you get this malabsorption.
Was it as lethal in cats as it is now in dogs?
I don't think anyone notices lethality in cats.
Sounds like a dog owner to me.
They just die. I think it probably was. Reading the more recent CPV strains may also cause clinical disease in cats. So I suspect that the original strain was also pathogenic in cats. But I don't know if people worry about it as much in cats. Does anyone know?
Well, look, if your cat's vomiting, has diarrhea,
that's right.
You notice.
Yeah, you do notice.
It's true.
You care.
So when you buy a pet, you should have it immunized.
That's normal
for
cats anyway. They're just throwing up a hairball.
Humans aren't the only ones that need vaccines. Your pets should be immunized.
Absolutely.
So you should have a rabies immunization. You should have a parvovirus. And we have a dog, and I'm sure she got both.
Sure.
So this is an interesting little virus. We'll reserve that story for Colin.
This is certainly, by the way, another quick little rat hole. This is a severely a first world problem, right? Our pets.
Yeah. I mean, in the third world, the animals run wild, right? And
what about the people? Just the people getting vaccines. You know?
Yeah.
This is true. Here we are worrying about the pets, but in many countries, the people don't even have the vaccines they need. And this comes up all the time. People email me and say, why are we worried about 2,000 people getting influenza when you have how many cases of malaria a year, Dick?
Oh, it's
millions.
It's in the billions, probably, unreported, but it's probably about 1.5 billion.
And dengue as well.
Oh, dengue is big time.
But it's all relative, and I mean, people are working on all of them. But the Parvo vaccine was very easy to make, and in animals, the clinical trials are non-existent. Basically, you just have to make sure it works. And
also, just you need to work on all fronts at once. You can't say just because there's a bigger problem, we shouldn't work on the smaller ones.
No, that's right. So small problems do have a way of becoming big problems after a while.
Yeah.
The reason I brought up this canine parvovirus is because I came across a paper in PLOS Pathogens, Public Library of Science Pathogens, called A Novel Boca Virus Associated with Acute Gastroenteritis in Australian Children.
Boca. Is that a term that perhaps means something?
I would guess it means mouth, right?
That's correct.
So these are parvoviruses, a new member. In fact, they're not even in this august textbook that I have in front of me.
Who wrote that textbook?
Well, this is not mine. This is Fields Virology, which is the encyclopedia.
That's why it's not in there, then.
It has every virus, but it's not here because this is a relatively new member of the parvovirus family. And these are the similar viruses. They're non-enveloped DNA, single-stranded circular DNA. And they were not too long ago suspected to cause respiratory illness in people. And now in this paper, they looked at people with gastroenteritis in Australia. They used genomic and bioinformatic techniques, and they identified a new variant of bocavirus in these people. It's different from the one that is causing respiratory disease. And here are some interesting numbers. So the most prevalent gastroenteritis virus, the rotaviruses and the noroviruses.
Right.
And then these bocaviruses are up there with them.
Interesting.
So they're in the stool of these people in Australia with gastroenteritis. So it's called human bocavirus species 2. And it's 23% different in sequence from the original Boca virus, which is causing respiratory disease. So I think now with this paper, which just came out in, I think it's April, this should stimulate more serosurveys of this.
Yeah.
Because gastroenteritis is a big problem globally.
Also,
I think it's interesting that two pretty similar viruses can cause very different diseases. The Bocas can cause gastroenteritis, and if you're 23% different, can cause respiratory disease. It's pretty interesting.
Isn't that one of the confusing syndromes that the coronavirus has caused?
That's right.
With the outbreak of SARS.
That's even more amazing to me because the coronaviruses are envelope. And how does an envelope virus survive in the gastrointestinal tract.
I'm not a virologist, Vince. I can't tell you.
Well,
neither am I. Maybe you could tell us, actually.
Okay, Vince, you're asking the question. Answer the question.
Envelope, non-envelope.
How does it survive?
Right, well,
it resists
virus,
digestion, you mean?
Yeah, an envelope virus has a membrane
Gotcha.
around it, which is derived from the cell that it replicates in.
Sure.
And non-envelope viruses don't have that. And envelopes are typically very sensitive, and that's why the viruses that have them, like influenza, are transmitted by respiratory routes, because they can't. So in your gut, you have high and low pH, you have proteolytic enzymes, and bile salts. This would wreak havoc on a membrane, right?
Maybe that's why we don't get infected that often.
But these coronas do infect the gut. And you know what? Influenza in birds is a gastrointestinal infection. It is not a respiratory infection.
And remember what I told you about Shope's?
Yes.
Shotes.
You know, the pH of the bird tract is probably low, right? There are probably digestive enzymes. I know it's a little different from the human tract.
It's a lot different, but you're probably right on that.
But in humans, flu does not replicate in the gut, by the way. No. And any GI symptoms are something else, probably another agent that's co-infecting you.
Intestinal flu, though. How many times have you heard that as a diagnosis?
That really bothers me.
It bothers me, too, of course.
Stomach flu or intestinal flu.
Yeah, that's right.
It's not. It's really bad to call it that.
Ain't no such thing.
There's no flu virus that will grow in humans, as far as we know.
Yeah.
But people use it on the news. They do it all the time.
They do.
Stomach flu. I would like to stop using that term, but it's too entrenched in the...
NIH wants us to even stop using the word swine flu.
Actually, Peter Pelletier said we should use swine flu to distinguish it from the other H1N1 that's circulating. And I would listen to Peter. You know what he told me about CDC? He said their job is not only to diagnose and find infections,
but to track it down and stop it.
But to manipulate the population in such a way to prevent spread of disease.
Right.
So in other words, they will not say, don't worry about this because it's like the H1N1 you had before, because then nobody will get a vaccine in the fall against it.
That's true.
Right. They would rather say,
I know,
previous infection with H1N1 is not going to help you with this virus, even though that may not be entirely true.
Yep. They're erring on the side of caution with regards to the outbreak possibility.
And it's important to point out, I mean, manipulating the population is not a bad thing in this case.
Right.
That's
yes. No, it just goes to show that they're not entirely scientific. They're involved in PR also.
Sure.
You wonder,
there could
Mark?
be massive conflicts of interest, though.
Yeah.
Right?
Yeah,
you know, who was the CEO of Gilead?
Donald Rumsfeld.
Right. At a time when bioterror was out and it was in the news.
Yeah.
No, but so if the government's involved in kind of or a government agency is involved in making a decision that is not truthful to the population, even though it's beneficial for the population.
Sure.
And there is a conflict of interest with respect to pharmaceutical sales. Right. All those great, sci-fi TV shows. There's always this conspiracy theory with a government contracted company. I have my company is funded by government contracts. We're a government contractor, and I have no conspiracy. I wish I had one too.
It can be arranged. I think Bernie Madoff has some good ideas here.
It's funny, at the beginning of Mark Crislip's podcast, he says, I have to tell you I have no conflicts of interest, although I would like to. Please send me money.
There's an honest man.
What did Donald Rumsfeld do, Mark? Tell us.
Oh, I don't know. That's all I know.
He was head of Gilead at the time, right? Previously, he was CEO of Gilead, which developed one of the two influenza neuraminidase inhibitors. And then when he was Secretary of Defense,
he recommended
the U.S. stockpiled the drug.
Of course, that's right.
No connection, I'm sure.
No, of course not.
In the
but
interest
it is a good drug. I don't want to deter people from Tamiflu. I think there might be here's that level of conspiracy, not a conspiracy, sorry, conflict of interest. When government official gets involved in pharmaceutical business, but it is a good drug, and I would not suggest to anybody that they not take it because it wasn't done right. It was done right. It's an excellent antiviral.
Absolutely. Don't get scared away from Tamiflu just because of Donald Rumsfeld. I just want to point out that the Boca virus is involved in respiratory disease was first discovered in 2005 in Sweden from people with respiratory infections. They used DNA methods and found this new virus. And now in the past year or so, it's found in the GI tract. So we'll, I'm sure, hear more about that in the future.
One last little interjection here. One thing that's important is that you try and cure the infection with the antiviral that you have, and not the one you wish you had.
Mm-hmm. Yes.
And of course, even better is to be vaccinated first. And if
sorry, that was a really bad Rumsfeld stretch there. You go to war with the army.
Yes.
I got it.
All right. Sorry, I
remember that. This convoluted conversation.
This is getting like car talk. Come on,
all right, guys. No problem. I'm sorry that we strayed from science, but we had a heavy week of viruses. We have a little heavy.
I just had an idea, though, Vince. Like car talk, they have these quizzes every now and then.
Yeah.
That's how they find out who's listening. We should come up with a quiz for our listeners.
Do you have a question?
No, but I'll think of one.
I think of one while we read email. Let's do some email. But before we do, let's have a word from our friends at ASM.
On May 17th through the 21st at the Pennsylvania Convention Center in Philadelphia, the American Society for Microbiology will hold its 109th General Meeting, the largest annual gathering of microbiologists in the world. Visit the General Meeting website at gm.asm.org to view the preliminary program, register for the meeting, or reserve your hotel stay. That's gm.asm.org.
Do you get a lot of email for futures, Mark?
Not that much.
But here's one from Key, QI. Hi, TWiV guys, or TWivers. I'm a fourth-year undergrad in microbiology, and I just want to say I love this podcast. I've listened to most of the episodes. The podcast is educative and understanding. I learned a lot and really appreciate your work. Right now, there is a swine flu outbreak in Mexico. I know what makes this new outbreak worrisome is that in all the recent cases, none had any direct contact with pigs, namely possible human-human transmission. For avian flu, people died, but no human-to-human transmission was shown. So my question is, once the virus can infect humans, what are the factors that prevent it from human-to-human transmission? E.g., why can't humans pass avian flu at this stage?
Great question.
It is the $60,000 question.
I actually know the answer to that, too, even though I'm not a virologist. Mm-hmm. The answer is that it turns out that the avian flu virus receptor, sugar, is a different sugar than is on the upper respiratory tract. It's only found in the lower respiratory tract, so you have to really breathe deeply in order to get this virus to attach. And that was a good study that was done. I forget the name of the investigators, but they showed the difference between these I want to call them isomers of the same sugar, and they probably are isomers, but there's another name that I'm looking for here that I forgot. Where the sugar is in this configuration, I'm indicating with my hands rather than in this configuration.
Well, what it is, Dick, is sialic acid is linked to galactose, which is the second sugar, either an alpha-2,3 or an alpha-2,6 linkage.
That's exactly what I was trying to say.
Alpha-2,3, I think, is the avian preference, and alpha-2,6 is the human. This is a good question because we can't study it very well in humans, right?
No, there's no model.
So for the viruses that have become able to transmit, we don't have the precursor. Like this, let's take this pig virus. If we could find the precursor to this one, which now can be transmitted, we could answer this question. You would have to do it in an animal model, though, right, of transmission. Well, ferrets or guinea pigs.
Sure. Are there cell lines of human epithelial lung tissue that you could use for this?
Yeah, but they don't answer the question.
Right.
You want to know transmission.
Okay.
Right. There is one paper, which I'll put a link to, which came out in March this year, which they identified 2 genes of influenza that confer avian influenza transmission through the air. So in other words, they took the 1918 strain and they made recombinants with avian influenza. They swapped genes, and they say in a ferret model, which allows it to transmit.
Right.
So they found 2 genes. These are the kind of experiments we need to do. Basically, we don't know the answer, but when you think about it, what do you need for transmission? You need to make a lot of virus in the respiratory tract.
Sure.
And you need to cough and sneeze to expel it. So any viral changes that stimulate those are going to improve transmission, just intuitively. Right?
Yes. But then there's another factor here with the avian flu virus, and that is the fact that wild birds are getting sick as well as domestic birds, and they're befriended by small children. Those little kids walk along the sidewalk or the streets, and they find a sick bird that it doesn't move as fast. It lets them pick it up. Mm-hmm. And they hold it close to them, and they start petting it and trying to make it come back.
What happens to those kids?
And they breathe in deeply.
They get infected. But do they transmit it to their friends?
They do not.
It's because something's missing, and we don't know what it is.
Okay, fine.
Is that clear, Mark?
Sort of.
Sort of.
We have to make sure you understand, because if you don't, our listeners are not.
We're missing our...
Basically,
I think it's fascinating. The biology is fascinating.
Yeah.
It's all about transmission from person to person, and these are viruses that typically are the birds are the reservoir for influenza viruses, the natural reservoir, and every influenza strain exists in birds, but it's not a threat to us unless it can go from person to person.
Right.
And the bird viruses don't do that. They have to undergo some changes, and we just don't know what they are at the moment. And we're just speculating that the kind of changes have to make the virus grow better so that when you sneeze and cough, you expel more of it. And it's a simplistic view, but has to be something else, too.
Sure.
I had a comment on Twitter this week. This gentleman, C. Forster, said, "Wow, there really is a podcast called This Week in Virology. So who's up for This Week in Literary Studies?"
Aha!
Well, I quote this here because the whole thing about the internet podcasts and the new media is that you can have very focused things like this, right? And you have a very dedicated listenership. So even if we have only 50 people listening, they're very interested in what we're doing.
Right.
As opposed to 1 million people, maybe, few of them care. Isn't that right, Mark?
Didn't somebody comment in the future everyone will be famous to 15 people?
That's right. That's right. Well, we have more than 50 listeners, but isn't that it, Mark? It's all about focused.
I think that's the amazing part, right? This is where you get really great quality. I mean, there's not every podcast out there is going to be great, but
we've proven that already.
There are at least one really good podcast for every topic that you're interested in.
Yeah.
Right. I love retro computing, so I listen to Retro Bits, right? And that podcast must have 400 or 500 listeners. And so it's not enormous, but everybody who listens to it loves that podcast.
Hey.
And same thing with This Week in Virology. I think there's going to be a lot of people that are thinking about starting a career in the life sciences, and they're going to start listening to this, and either they're going to love it, and they'll follow up on it, or they'll see, well, it's maybe not for me, but those who love it will really appreciate your show. And I certainly think with the ratings, it's certainly. Right now is an amazing time to have This Week in Virology.
It's perfect, right? Yeah.
We're in the hot zone.
Well, the thing is, there will always be outbreaks, right?
Right.
And if you can convince a few people every show to, go get that vaccine this season, you're actually going to save lives. So this is the podcast that saves lives.
That's
right?
a good way of looking at it.
I'm glad we didn't say that, but I'm glad you did say that.
It should be our tagline, right?
It's true, though. Yeah.
Yeah, you're right.
This is
right.
What's your tagline? The kinds that make you sick.
Yeah,
that's right.
Well,
maybe we should take the other route. That's a good point. Okay, here's an email from Angelo, who I know, and he's actually going to be a PhD student here in the fall.
Nice.
I was completely unaware and fascinated to learn that highly pathogenic strains of flu contain a unique ORF open reading frame within the PB1 segment. A quick scan of the literature, and I was able to learn that this PB1-F2 protein is translocated to the mitochondrial membrane where it punches holes, resulting in cytochrome C release, thus leading to apoptosis. This is very interesting because how this would result in greater fitness for the virus is not clear to me. For obvious reasons, a virus does not want to kill its host, but in the case where there is cell lysis taking place, it might make sense if the virus is spewed into the extracellular space. But if PB1-F2 is inducing apoptosis, this shouldn't result in cell lysis, so the virus is effectively killing its host cell, but not reaping the benefit of spreading. So he wants to know that. On a separate note, I was surprised that you didn't mention Peter Pelletier was your former PhD advisor. He was talking about my conversation with Peter on Wednesday. Well, I think I've talked about that quite a bit, so I didn't think. But anyway, this PB1-F2 is a little overlapping 80 or 90 amino acid protein made from one of the flu RNAs. And Peter Pelletier has shown that in very pathogenic strains of flu, this protein is made and has a very specific amino acid at position 66. And if you change that, it makes the virus less pathogenic in an animal model. So guess which viruses have this protein with the pathogenic amino acid? 1918 pandemic, 1957, 1968. The H1N1 from swine.
Missing.
They don't even make the protein. Not all strains make it. And in fact, the other H1N1 that's circulating also doesn't make it. So that's why he thinks this is not going to be a terribly virulent
How about the avian
virus.
flu virus, though, H5N1?
They have them, yeah.
They do have
They do have them.
Okay.
This gets back to my point from last week that if this was engineered as virus was engineered as part of some conspiracy, that
Alan,
they did a
you're
really
quite right on
crummy
this one.
job.
That's right, Alan. They would have put in PB1F2 with the right
Yeah.
amino acid.
It's
Yeah.
good enough for government work.
Yeah, right.
Gentlemen, please, we're dependent upon them for our livelihoods. Indirectly, but nonetheless.
This whole question of whether a virus wants to kill its host or not, this is really interesting. I want to hear your takes, but my view is if the host lives long enough to transmit infection, it doesn't matter if it's dead or not.
The biology is fulfilled.
Right?
Right.
Yeah, it has to fulfill its biological destiny, whatever that is.
And, the whole idea of a virus becoming more some people don't like this idea that it would evolve for more virulence, but, again, if you live long enough to transmit, or if you cough or sneeze harder, which is increased virulence, you transmit better.
True.
And it depends on how the virus is spread. If you've got a virus that is spread by person-to-person contact through aerosols, then it doesn't want to make the host so sick that they can't get up and go and meet susceptible hosts.
Right.
But if you have a virus that's spread by mosquitoes, then incapacitating your host and putting them in a coma and killing them rapidly is probably a great idea, because you attract more. They can't swat away the mosquitoes that are going to feed on them and carry the virus someplace else.
That's a good point.
The evolution can drive it in either direction.
We're running an enormous risk here of anthropomorphizing this whole thing.
Yes.
Viruses don't want to do things, and nature doesn't want to do things, but it's driven in a certain direction because of selective pressures, right? So in this case, this virus has infected all age groups. So it can kill randomly. It doesn't matter. Even if it isn't transmitted by the fact that it's not long enough lived in, let's say, somebody who just died from it. It doesn't matter because most other people get asymptomatic infections or mild infections. So deaths from viruses. I mean, the only one that I know of that's really super lethal that I would say what's going on here is rabies. But that's not our virus. That's a bat virus.
Right.
So even
I have a virology question for you guys. And this is another tangent. I'm sorry for the
okay.
It's okay, no problem.
The human cytomegalovirus?
Yes.
Okay.
It has a couple of genes, US2 and US11. There's probably several others, but what those gene products do is they sort of attack the major histocompatibility class 1 heavy chain production, the MHC class 1.
Right.
So it has an active strategy to prevent, the display of viral proteins on the cell surface and prevent the immune system from. Is that like an unusual virus, or do most viruses, besides just replicating and getting out there, do they go in and tinker with the cellular machinery to prevent the immune system from working?
Well, most viruses have some way of antagonizing.
Yeah.
So, you're talking about the adaptive immune response. There are dozens of incredible strategies, all along the MHC pathway. And then there are strategies at the innate immune response as well.
And interleukin inhibition.
Interferon and cytokine production.
That's right.
Bottom line, Mark, is that the immune response is so effective that unless viruses evolved to antagonize it, they would never survive, I think.
Right.
Oh, okay.
So it's more the rule than the exception. It's not unusual.
Yes.
Dozens of incredible mechanisms.
That's right.
And those things actually teach us a lot about the pathways.
Yeah, in fact, one of the things you want to know if you find a new virus is, well, how does it deal with the immune system?
Indeed. Or how come it just arose?
Right.
This is Boca-virus. It's fascinating. Right? 2005, here it is. In 2004, where was it?
In fact, someone asked us to talk about evasion mechanisms. We should do a show. It could do many shows, but we should at least summarize it at one point.
Or we could just keep dodging the topic.
Well, we get
how appropriate.
We get distracted by outbreaks.
Yes.
Indeed.
I'm going to read one more email because this is someone who came from TWiV, and that's a consequence of your help, Mark. This is from Richard, who says, "Hi, guys. Thanks for the podcast and for making it accessible to the non-scientist. I jumped host, as it were, from TWiV, Security Now, etc. I'm interested in your podcast because I work in the sewage treatment industry, and it is something that has always worried me. Right. I know quite a bit about sewage treatment and often have to look at sludge to check on the different types of protozoa and filamentous bacteria. However, I know next to nothing about the virus. I'm learning about them thanks to you." Yeah, sewage is a huge source of enteric viruses, these gastroenteritis viruses, polio virus.
Yeah.
You know anything about sewage, Dick?
I know a lot about sewage, actually, Vince.
Really?
Yes. And why do I know about sewage? Because a lot of the parasitic diseases that are helminths, worms, have stages which survive for long periods of time in sewage and in sludge. There's a layer that develops in some of these filtration things called Schmutzdecke. It's a German term. And the Schmutzdecke has...
If you have any complex issue, leave it to the Germans to have a word for it.
That's right. They have a word for this. So Schmutz is like, dirt, and Decke is a layer. So a dirt layer, that's actually what that translates out to. And if you look in that Schmutzdecke layer, you can find Ascaris and Trichurus eggs that are over 10 to 12 to 14 years old. I mean, they've survived a long time in this environment. So we've had to know about this from my past life as a parasitologist. Yeah.
The sewage is a wonderful way of spreading viral infections because, as you know, we excrete viruses in the feces.
Absolutely. It
goes in the sewage, and unfortunately, sometimes sewage contaminates oyster beds, and that's how you get hepatitis A, and there are other examples. So we'll keep that in mind and educate you, Richard. But that's good. We had a couple of people come over from Twit, and welcome to all of you.
And by the way, I just saw that the domain name schmutzdecke.com appears to be available. So,
darn. All right, let's go for it.
All right, gentlemen, let's have some picks. Any science pick. This is something new here. We'll start with Mark. Just something to do with science that our listeners would like.
Well, this pick is free, and it's an application called Celestia. That's CELESTIA. And what it is a simulation. It's a 3D simulation of our universe.
Oh, wow.
And it works on Mac, it works on PC, and works on Linux. It's open source. I think there's 3 million users right now. And so you can imagine, so when I first downloaded it, I must have spent 2 hours in this 3D simulation, just basically flying it throughout the universe.
Nice.
I think there's 100 solar systems. There is, an incredible level of detail within our own solar system. So if you want to go catch up with Halley's Comet, you basically either fly to the comet, or you can use their browser. There's like a little browser menu option where you just click, okay, go to Halley's, and then it'll fly you up to about 100 miles off Halley's Comet. It's scientifically accurate, right? And it's updated all the time.
Who manages it?
Celestia.
No, is this a group of astronomers at the Arizona State University or something like that?
Let's see. I didn't go into that much detail.
Right, because I know Google
6,000 members. 6,000 members that manage it.
That's cool. Because I know Google has a universe type surveying device also. Yeah,
this is 10 generations ahead.
Neat.
This gives you an incredible level
Celestia.
of a sense,
right?
You can fly right past the moon. There's also these tours which you can download. So you download the little tour, and it's a script that runs inside the program. Very easy to use too. My 4-year-old at one point, he's 6 now, but when he was 4, he spent about a half a year, 45 minutes a day, exploring the universe. And his fun thing to do was to find exoplanets, right? So he'd click on a star, he'd fly up to that star, and then look for an orbiting planet.
Cool.
And then he'd write it down, he'd take a screen cap of it, and then he'd go to the next.
Good heavens.
It's a lot of fun.
Sounds great.
You got to try it. They have the so these scripts, by the way. You run the script, and it'll take you through the pathway of a spaceship, an Earth ship that's gone like Voyager, and it'll do all the ricochets through our solar system the same way it did, but in a very accelerated time.
Gotcha.
And it'll, the Cassini mission, it'll fly you around the Jupiter, then down into Saturn.
Yep.
Cool.
Definitely a lot of fun. So it's free. Every computer should have one installed. Every family computer, because if you want to get your kids into science, this is a completely other end of the scale here. We're talking about viruses. Now we're talking the scale of the universe. And you can you can pick a star that's in an arm in the Milky Way galaxy, or you can even go farther. You can fly out of the Milky Way galaxy and see our galaxy from a different perspective. So my pick is Celestia, and I think it's celestia.org. I just Google it. I don't even bother typing the website. Just Google Celestia, you'll find it.
That's great. We do a lot of astronomy recommendations here because it's a great science. So that's beautiful.
Exactly.
Dick, what do you have for a weekly pick?
Well, one of my all-time favorites that I go to regularly is the USGS website. That's United States Geological Survey, and they have always done very well with maps and always done very well with disease distribution maps. And in fact, I think in an earlier episode I described how they were established.
Mm-hmm.
And it involved mapping out where hookworm was found in the United States.
Great.
And it follows soil types. So once they discovered that, they said, well, why don't we just soil type the entire country and see if other diseases fall out like that as well. I'm not sure any others did, but the USGS was up and running at that point. So I love their maps of disease distributions. And the CDC maps are good, but the USGS maps are really great.
Mm-hmm.
So that's my favorite site.
Very good. We'll check that out. USGS website.
Yep.
Alan.
.gov. It's .gov.
.gov.
.gov, yes.
Alan.
So my pick of the week is one that comes up occasionally in my own work as a science journalist. There's this group called the Society for Amateur Scientists.
Gee, that's all of us.
Yes, exactly. They were founded a number of years ago. And I remember when they first started going, it was about 10 years ago, I think. And I thought, wow, that's a really cool idea that they've got this organization for people who are just into science, who don't necessarily do it professionally, but they enjoy it. And this predates, Make Magazine and the whole revolution in
Scientific American
do-it-yourself type stuff. And yes, it was. It sort of grew out of Scientific American, which has obviously been around for a long time. Yeah. But the website is sas.org, and they are kind of a meeting place and clearinghouse for information for people who are into science to the point that they want to do some of it on their own. There's a fair amount of information. For kids working on science projects, which is obviously a big audience. But the real focus of the organization is grown-ups who, want to play around with backyard astronomy, for example, which kind of ties into Mark's suggestion. There's, I would say, thankfully, there's not much virology in this group.
That's right.
But there's a lot of interesting biological research that people are doing, that amateurs are doing these days. I mean, you've got your usual backyard bird watchers and that sort of thing, but there's also now a real trend toward people just playing around with bacteriology. They'll set up a few petri dishes in the kitchen. And you can go to a number of suppliers for various harmless organisms to culture. By the way, for listeners who are thinking of doing this, I would suggest that you get a culture of something is harmless if you're going to play around with it.
You should at least buy Bergey's manual.
Yes, exactly. Kind of know what you're working with, because there is some bad stuff that you could just culture out of the soil.
Yeah, most of it's in my refrigerator.
Yes, sorry.
This is kitchen top biology. I understand it's gaining popularity.
Yeah, and so SAS is one of the organizations that promotes this sort of thing, and I think it's a really positive trend.
Cool.
Nice.
Very good. Society for Amateur Scientists. Yes, we all are, aren't we?
Yes.
All right, my pick is a online game called Sneeze, and if you go to it now, you're going to see the splash screen is in fact someone sneezing and spraying green goop all over. This is developed by the Wellcome Trust. It's not nonsense, okay? No. It helped to prevent the spread of swine flu by catching your sneezes with a tissue and then washing your hands. This educational software game illustrates the consequences of allowing flu to spread in an interactive graphical simulation. It is intended to inform people of the reasons to take measures that will help prevent further spreading of flu. Where did I find this? Oh, my colleague Saul Silverstein sent it to me the other day, and I showed it to my kids yesterday, and they're just playing it continuously. It's extremely simple. You go into a room, you're an adult, and you can spread it to children, middle-aged, or older people, and each of them have a different susceptibility, which is explained. And the goal is to get more and more people infected.
But of course.
And then you move up levels beyond, and kids are the best spreaders, so you want to sneeze on a kid.
That's right.
It's very simple, but it's addictive, and it's kind of relevant, so It's a lot of fun. Just check it out.
How do you sneeze on it? I've got it loaded up there.
You space bar. You hit the space bar and you use the arrow keys to move the guy around. Because you want to make sure you're near a lot of people when you sneeze. You don't want to sneeze on the subway car.
So Vince, are they going to go to a sexually transmitted disease scenario soon?
Dick, this is a family show. We can't get
That's
that.
one of my picks for the week, actually.
Okay, thanks for that. That's good. I like these picks. Maybe we'll do this for a while.
So in this game, the flu is green and slimy.
That's too bad.
That's too bad. But it's just so you can see it. When someone sneezes, they have great sound effects. And every now and then someone will go, oh!
They snort and all that stuff. Oh, jeez.
It's really fun.
Isn't that funny?
Oh, brother.
Yeah, so check that out. Okay, those were our picks. And I forgot to say, I think we should tell people if you want to send in questions as audio files, you could do that. Send us an MP3 file. We'll play them on the show and we'll answer them.
Yeah.
Okay, so record it on your laptop or computer, make an MP3 file, email it to...
Or the questions of the week.
TWiV@twiv.tv.
We'll put it out to the general public
and we'll
for
do
answers.
questions, too. So, thanks, everyone, for doing this. Mark, thanks for joining us. That was great.
No problem.
Hope you enjoyed it.
We enjoyed having you,
fun.
Mark. It was fun.
It was a lot of fun.
So
I learned a lot.
Where do you want people to reach you?
Well, they can visit twit.tv /FIB, FIB, for Futures in Biotech. And I'd like to make an announcement that, Vince, you're coming on the show next week, which is May 15th.
Nice.
And we will be recording live at 4 p.m. Eastern Time. That's New York City time. And this is going to be a panel show, and we're going to talk about the most amazing scientific stories in the past probably about 10 years. So we're going to have 4 panelists and myself, and we're going live. So you can watch online or download it later on. But so that's May 15th at 4 p.m. Eastern Time, and that will be at live.twit.tv.
Well, you sound good. He's got a good mic, doesn't he?
He does. The other thing you can tell right away is that Mark is from Canada.
Oh, yes. He's Canadian.
Really? What's that all about?
Well,
He's from McGill, you know. You went to McGill, right, Mark?
That's right, yeah.
Wonderful.
Yeah.
And I actually collaborated with a Pelletier years ago here.
Uh-huh.
So he said it's a very common name. It's very interesting.
There's 10,000.
I've got a French name, and I didn't go to McGill.
Despommier, yeah. Mark, hope you'll come back sometime.
Thank you very much for the invitation. I appreciate it. It was a lot of fun.
Yeah.
Alan, thank you.
Thank you. It was always fun.
You're at dovedocs.com, right?
Yep.
And Twitter is Alan Dove.
Right.
And Dick is at verticalfarm.com, right?
I am, but I'm also at one other thing that I'm sure your listeners and my listeners would like to know about, and that's what I used to work on, called Trichonella.
Mm-hmm.
So trichonella.org. They can go to that website and learn all about Trichonella.
And you bought that domain name.
I did.
You should hang on to it. It's a good one.
I'm going to.
Can they learn all about parasites there?
They can learn all about Trichonella there.
All right. And do you have a podcast on Trichonella?
I don't.
That would be focused.
That would be very focused. It's all about eating pork tartare. All the recipes for pork tartare.
Well, this podcast has a website, twiv.tv. You can send your questions and comments to twiv@twiv.tv. And you can follow me on Twitter at PROFVRR. We will be back next week.
Indeed.
And then the week after, we will be live at ASM.
We will.
Which will be fun. So please come back next week. You can find us at twiv.tv or on iTunes, and you can subscribe and get all the updates right away. You've been listening to This Week in Virology, the podcast about viruses. And what was the tagline?
The kind that make you sick.
Yeah, but we had a new tagline.
The podcast that saves your life.
The podcast that saves
that could save your life.
We have to use Mark's voice is perfect.
It is,
Mark,
absolutely.
Do the tagline. You've been listening to do that whole spiel.
This Week in Virology, the podcast that saves lives.
It's just mellifluous. It's great.
Beautiful.
Thanks for joining us. See you next week. Another TWiV is viral.